Lycopodium belongs to the division Pteridophyta and the order Lycopodiales. The order includes slender, weak-stemmed plants with small, moss-like leaves without ligules and club-shaped fruiting bodies or strobili. Antheridia are of embedded type with biflagellated antheridozoids. The order with a single family Lycopodiaceae comprises two living genera Lycopodium and Phyloglossum and the fossil Lycopodiates. The Lycopodiums are familiarly known as ground pines, trailing evergreens, club mosses. Now, the distribution and habit of the Lycopodium. Lycopodium is worldwide in distribution and possesses about 400 species worldwide. A large majority of the species are found in tropics. Plants are slender, herbaceous, or shrubby sporophytes with a wide range in habit and habitat. In tropics, there are mainly the pendulous epiphytes, and the temperate ones are prostrate trailing forms with upright branches, and others are erect plants. All species have branching rhizome creeping above or below the surface of soil from which arise aerial branches. Aesthetically more valued are pendulous epiphytes. Now we will discuss its morphology. The plants are rhizomatous herbs. Stems are clothed with small, simple, sessile leaves of broad base and single unbranched vein. They range from minute scales to broad leaves 2 to 3 centimeters long. Leaves are mostly entire, but in a few are serrate, as in Lycopodium serratum. In most species, leaves are spirally arranged, but some species show whorled or decussate arrangement. The branching is fundamentally dichotomous. Roots are adventitious, occur on the undersurface of the stem. The genus has been classified into 2 subgenera. Members of subgenus Rhodostachya are terrestrial species with prostrate creeping axis and erect branches. The adventitious roots occur at the branchings. The first branching may be dichotomous, but later branchings may appear more monopodial, unequal dichotomy. Sporophylls are with dentate margins, are smaller than foliage leaves and form strobili, which are simple or branched. Species including the subgenus do not form bulbils. In another subgenera, Eurostachya, the plants do not have creeping axis but are erect if terrestrial or pendulous if apathetic with the dichotomous branching. The adventitious roots emerge at the basal region only. The sporophylls are alike to foliage leaves and are restricted either to distal end of the stem and branches or are present on alternate zones of fertile branches, and sterile leaves are present all along the stem and branches. Plants generally form bulbils. In Lycopodium, growth takes place by a group of meristematic cells rather than a single apical cell as in stems of most pteridophytes. In dichotomous branching, single center of meristematic activity gives rise to 2. The initiation of unequal centers is common and results in pseudo-monopodial branching, as in Lycopodium densum and Lycopodium obscurum. The most unequal branching results in the formation of condensed axis, which becomes specialized for vegetative propagation and are called bulbils or gami, which on falling from the parent plant secure vegetative propagation. The factors favoring the formation of bulbils are not known. Histologically, the stem has one-celled epidermis with stomata, a cortex, and a central vascular cylinder. Relative thickness of the cortex and stele is variable in different species. The cortex in some species remains parenchymatous, and in others with age the cortex gets sclerified into outer and inner zones, forming 3 bands, and in extreme cases the entire cortex gets sclerified. Long aerial stems normally has 3-zoned cortex: hypodermal chlorenchyma, followed by parenchyma and sclerenchyma encircling the stele. In subterranean stem, the cortex is often completely sclerenchymatous. Aerial stems often have large air spaces in cortex due to disruption of cells. Endodermis is usually ill-defined and can be identified in younger portions due to thickened radial walls, that is known as Caspian strips. Internal to endoderms is pericycle layer of 3 to 6 cells. Stelar anatomy differs from species to species and even within the single species. However, young plants in all the species conform to a single pattern: a single rod of xylem and radiating arms and phloem in between xylem arms. Now, xylem and phloem are separated from each other by a layer of stellar parenchyma. Some species even retain this simple satellite arrangement with xylem of only 4 arms. In a deviation from this basic pattern, irregular and increased furrowing of xylem takes place, and in a cross section it appears as isolated strands, as in Lycopodium lucidulum. The furrowing may result into plate-like lobes, as in Lycopodium volubile. In some forms, the xylem retains its radiating arms pattern but also has patches of xylem within the benxylem, separated by parenchyma or strands of phloem, as in Lycopodium squarosum. The Steele's exarch. Wilden 1970 has, however, found the mesoxylem in Lycopodium clavatum, Lycopodium tristium, and Lycopodium lucidulum. The protoxylem elements form a peripheral group of up to 20 cells with few or no intermixed parenchyma cells. This feature separates Lycopodium from all other living vesicular plants. Their secondary wall thickenings consists of indirectly connected rings. Metaxylem tracheids are either scalariform or have circular bordered pits. The sieve cells of the phloem are elongated and pointed. The sieve areas are scattered and are inconspicuous on lateral and end walls. The embryonic root is short-lived. The roots on older plants are adventitious and show equal dichotomous branching. Over the apical meristem of the root is the calyptra, and proximal to the apex is a long zone of unicellular hairy extensions borne in profusion. The cortex in roots with a complete tissue maturation is often completely sclerenchymatous. The xylem is crescent-shaped with phloem in the concavity. The roots are often diarch. 2 protoxylem points are the 2 poles of U and C-shaped vascular strand, but often 1 or more protoxylem points are also located on the convex side of the stele. The former matures earlier than the later. The roots originate chiefly from pericycle tissue of the stem and do not grow out straight through cortex, but grow vertically through the stem cortex before emerging below. As a result of transection of stem, many roots are seen in the cortex and are oriented in such a way that the open ends of their xylem face towards the outside of the stem. The roots of Lycopodium are peculiar in 2 respects. The lateral ones are not endogenous in origin, and the root hairs are paired. In some species, root stele is very similar to the stem stele. At the point of attachment with the rhizome, the root is polyarch and exarch. Therefore, except for size, it becomes difficult to distinguish the 2 organs. Lycopodium reproduces vegetatively by various propagules such as gemi or bulbils, body fragments, formation of resting buds, formation of root tubercles, and by the formation of adventitious buds. Asexually by spore formation, and sexually by the formation of specialized sex organs. Now we will discuss these reproductive methods one by one. Number 1, vegetative propagation. It occurs by the following methods. Number 1, by the formation of gemi or bulbils. These are modified vegetative structures that arise as lateral outgrowths from near the stem apices and take the place of leaves. They have been reported in Lycopodium lago, Lycopodium phlegmaria, and Lycopodium lucidulum. Each bulbil consists of a short and reduced axis surrounded by a number of thick and fleshy leaves. These leaves store food material. They remain on the plant till a root primordia appears on the shortened stem. The gummi fall on the ground and grow into a new young plant. Now, number 2 is fragmentation. Death and decay of the older regions of the stem leads to the separation of younger branches, which grow into separate plants. This is the most common method of vegetative reproduction in Lycopodium. Now, the formation of resting buds. In a Lycopodium undulatum, the tips on the apical buds of the rhizome and its branches store food material and become surrounded by densely crowded leaves, forming the resting buds. In winter, the whole plant except these resting buds die off, and at the advent of the favorable conditions, the resting buds resume growth and give rise to the new plant. By the formation of root tubercles. In Lycopodium ramulosum, the tubercles originate from the parenchymatous regions of the root cortex. They consist of a group of cells which store food material and protected by thick walls and have the capacity to germinate into the new plant. Now we will discuss the asexual method of production in Lycopodium. The asexual spores are formed within the sporangia, which are all alike, homospores. The sporangia are borne on the upper surface of sporophylls. These sporophylls are borne at the distal end of main axis or branches to form strobili or cones. The cones rarely may be borne on the long stalks with scale-like leaves. The sporophylls are smaller than foliage leaves, and in some species they are chaffy scales with dentate or serrate margins. In pendulous epiphytes, the strobili are often highly branched. In strobili of Lycopodium drummondii, the apex becomes active and forms a vegetative axis with reduced leaves, which in turn matures into a strobilus. In some species, for example Lycopodium lucidulum, where cones are not formed, sterile and fertile leaves from alternate zones all along the stem, and in some, all leaves in a mature plant form sporangia. Such species are considered to be primitive and are included in Slago group. These species characteristically have undifferentiated strobilus, closely similar vegetative leaves and sporophylls, and the entire metaxylem elements consists of circular bordered pits. Each sporophyll produces only one sporangium. The sporangium is borne on the upper adaxial surface of the sporophyll, but later may become axillary. The sporangia are 1 to 2.5 mm in diameter, reniform to subspherical, with short stalk or pad-like base. The mature sporangia are yellowish and liberate spores through a slit in a wall transverse to sporophyll. In many species, the sporophylls fold backwards, exposing the sporangia as they mature. Sporangial initials differentiate normally on the upper side when sporophyll is still embryonic. Therefore, in a longisection of the developing cone, all stages can be seen. By periclinal division, the initials form inner and outer layers of the cells. The mature layers form sporogenous cells, and the outer layer forms the sporogenous stalk and the wall of the sporangium. This process, in which sporangia develops from a group of superficial cells, is known as eusporangiate method. After periclinal division, due to the differential growth, that is, the active divisions in the sporogenous tissue, results in upward bulging and the sporangium assumes a reniform shape. Later, periclinal division in surface layer lead to the formation of 5-celled outer layer. The layer surrounding the sporogenous cells stain deeply and acts as tapetum and gets decomposed during maturation of spores. As the sporangia mature, the peripheral layers also break down and the mature sporangium has only one cell thick wall. Due to the breakdown of peripheral cells and the tapetal layers, spores come out and come to lie in a fluid mass. The spores which are produced in a large quantity are homospores. That means all the spores are identical, small, light, with smooth or ornamented walls, and a triradiate mark, a point of attachment in spore tetrad. They contain oil as reserve food material, and in some species are chlorophyllous. Now we will see the spore germination. Spore germinates in few days after dispersal and forms small, free-living green protheli. Such protheli are cylindrical or ovoid with branches or lobes and are somewhat short-lived, maturing in the same season. In other species, the spores are non-green and thick-walled. The germination is delayed by years, and in the meantime, spores get buried and form subterranean mycorrhizic protheli which are non-green. To begin with, such protheli are top-shaped and later becomes irregular or convoluted and takes years to mature. The former type, surface-living and green protheli, is prevalent in tropical species and later subterranean and non-green in temperate creeping ones. The gametophytes very early in their development, at just 4 to 5 cell stage, come to have a mycorrhizal association. Further growth takes place by a group of meristematic cells organized at the apex. Gametophytes in some species are partly subterranean with chlorophyllous aerial portion. In some epiphytic species, for example in Lycopodium phlegmaria, the prothallus is irregularly fragmented and these fragments form gamete which secure vegetative propagation of the gametophytic generation. In Lycopodium, the sexual reproduction is oogamous type. The sex organs are present at the anterior end of the protheli. The protheli are monoecious and each sex organ originates from a single superficial cell just behind the apical meristem. In subterranean types, the Antheridia and Archegonia form distinct patches and cover the entire crown or the base of its lobe. In elongate types, they are present on the central cushion and are intermingled. Antheridia are sunken and produce a large number of pear-shaped biflagellate anthrodiosids, which are attracted chemotatically by the archegonial exudate citric acid. Archegonia are sunken at the venter and only the neck protrudes. Subterranean protheli have archegonia of long necks, as many as 14 neck canal cells, as in Lycopodium campanillatum, and surface-living protheli have short necked archegonia, sometimes just one neck canal cell in Lycopodium cernum. At maturity, the neck canal cells and the ventral canal cells disorganize, creating an open passage for the anthrodiosids to enter the neck and reach the egg. Now we will discuss the development of antheridia. The protheli are normally monoecious, but the antheridia usually develop first. The antheridia originates from a single superficial cell just behind the apical meristem. This is known as antheridial initial. It divides by a periclinial wall into an outer and inner cell. The outer cell, known as the primary wall cell or jacket initial, divides by repeated anticlinal divisions and forms a jacket layer, one cell in thickness. In surface view, there appears a small triangular cell in the middle of the jacket layer known as opercular cell, which marks the point of exit of the anthrodiosids. The jacket layer in Lycopodium phlegmaria is two-layered towards the periphery. The inner cell, called the primary androgonial cell, by successive divisions forming a large number of androgonial cells. The androgonial cells further divide to form anthodiosid mother cells or androcytes. The metamorphosis of the androcytes into anthodiosids has not been followed in detail, but According to Lang 1988, it resembles with that in other vascular cryptograms. The mature antheridia vary in size, shape, and in the number of antheridioses in different species. They are entirely or in part sunken in the tissue of the prothallus and consists of oval mass of androcytes projecting slightly from the prothelial tissue and surrounded by a wall, which is partly formed by the jacket layer, developed from the jacket initial of the young antheridium and partly from the prothellal cell or prothellal tissue. The antheridiozoid may be an oval cell about twice as long as wide, with the anterior end somewhat pointed and bearing two long flagella. For example, in Lycopodium clavatum, or it may be elongated and somewhat twisted with two flagella, for example, in Lycopodium phlegmaria. In a Lycopodium clavatum, the nucleus lies on one side of the cell, and the cytoplasm forms the greater part of the antheridiozoid. The antheridiozoids are less specialized than those of other pteridophytes and are much like those of bryophytes. Now we will see the dehiscence of the antheridium. When the antheridium is mature, either the single triangular opercular cell or some cells of the jacket layer become mucilaginous, and then the androcytes absorb water and rupture the antheridium. The archegonial initial is superficial. It divides by a periclinal wall into an outer primary cover cell and inner central cell. The primary cover cell divides by two intersecting walls into four neck initials, which divide further by transverse walls to give rise to the neck that varies from 3 to many cells in height. The neck is composed of 4 longitudinal rows. The central cell divides by transverse wall into an upper primary neck canal cell and a lower primary ventral cell. The former may function directly as a single neck canal cell in the archegonia of aerial protheli, or it may divide repeatedly by transverse divisions to form a variable number of neck canal cells. In a Lycopodium slago, there may be up to 7 neck canal cells. In Lycopodium campanillatum, there are 14 to 16 neck canal cells. The primary ventral canal cell undergoes a transverse division to form a lower egg cell and an upper ventral canal cell. In some cases, the primary ventral canal cell does not divide at all and becomes slightly broader and functions directly as an egg cell. In the mature archegonium, the neck portion protrudes out while the venter remains sunken. The extra row of an archegonium consists of an egg and a varying number of neck canal cells and a ventral canal cell. At maturity, the neck canal cells and the ventral canal cells disorganize, creating an open passage for the antheridiozoids to enter the neck and reaches the egg. Now we will see the embryo, the new sporophyte. First division of the fertilized egg is transverse to the long axis of the archegonium. The outer suspensor cell normally does not undergo any division or may become 3-celled. The inner cell gives rise to 2 superimposed tiers of 4 cells each. The upper tier in some species swells up to form a foot, and from the lower tier is derived the embryo proper. 2 cells facing meristem develop into stem, and other 2 forms leaves and primary root. Root differentiates laterally at a place where apical region and foot are joined. In some species, Lycopodium serenum, distal tier develops into a massive globose structure, protocorum, and pushes its way away through gametophyte. The protocorum bears rhizoids and from its upper surface differentiate leaf-like avascular structures, that is protophyls. After the formation of many protophyls, a shoot meristem is organized on the protocorum. The first root arises from the base of the stem. Protocorum, because of its structural organization, apparently intermediate between gametophyte and sporophyte, has been looked upon by earlier workers as evolutionary forerunner of leafy vascular plant.